Hi everyone. In December 2025, I had the pleasure to attend a chemoinformatics meeting in Paris, organized by the French Society of Chemoinformatics. The event brought together researchers from academia and industry for keynote lectures, short oral presentations, and a poster session. But before we get into the meeting itself, I'd like to briefly introduce the French Society of Chemoinformatics. The French Society of Chemoinformatics was created around 2007, following an idea first put forward by my colleague and friend, Professor Alexandre Varnek. The society promotes chemoinformatics as a discipline in its own right, at the interface of chemistry, informatics, data science, and artificial intelligence. It brings together a community of researchers, students, and industry scientists, and thus federates members across academic labs and private companies. The society supports the community through scientific events designed to connect academia and industry, providing researchers with a platform to present their work. It also serves as a space to discuss careers and scientific strategies in the field, both nationally and internationally. The Society offers mobility and conference grants to help students and early career researchers participate in training or short research stays. On a more personal note, the Society is especially meaningful to me. Back in 2007, I was awarded a crucial grant from my institute, the French National Institute of Health and Medical Research, which was instrumental in launching the project. I'd like to take this opportunity to warmly thank again the INSERM leadership team at the time for supporting the structuring of our scientific community. And I also had the chance to contribute as president around 2010. More recently, the society was chaired by my colleague Professor Mathieu Montes, and the presidency passed to Professor Gilles in mid-December 2025. Back to the meeting. A few keynote lectures really set the tone. From Evotech, the presentation explored how in silico science meets medicinal chemistry in real-world industrial settings. The presentation from Servier discussed how AI is reshaping drug discovery, particularly with the rise of agentic and conversational AI in research workflows. We also heard about targeting RNA with small molecules from Qubit Pharmaceuticals and advances in AI for chemistry from the University of Strasbourg. In this short video, I won't cover everything as it would be too long. Instead, I'll focus on a small selection of new computational tools presented in talks or posters, especially those that are open source or are expected to become soon accessible through GitHub or related repositories. Let's start the tool highlights with something very practical: chemical space visualization. Spherical generative topographic mapping is a method that maps chemical structures onto a sphere instead of a flat surface, helping avoid artificial edge effects and better preserving the global topology of chemical space. The Andromeda tool was then presented. It is a new drug discovery library that centralizes chemical, biological, and computational data in a secure local web application designed to improve collaboration across disciplines, especially in academic labs where existing solutions can be limited or very expensive. Next, a contribution on fingerprints. The author presented a deterministic algorithm to reconstruct molecular structures from extended connectivity fingerprint vectors, a representation often considered non-invertible. The next study introduced an AI-powered framework that combines chemical space visualization with collaborative language model agents to, for instance, retrieve data, build interpretable maps, and analyze structure-activity relationships, among others. In the Code2 Smell study, researchers built a database of 75,000 experiments showing which smell receptors respond to which odorant molecules, then combined protein language models with graph neural networks to predict receptor activation. On the binding site side, another highlight was a comprehensive open-source databank derived from the Protein Data Bank 2025, extracting over 300,000 pockets with structural and physicochemical descriptors and offering a web interface to filter and download binding sites tailored to specific needs. On the docking side, another tool is ARIA Dock. This is an automated, active learning docking workflow. It docks an initial, diverse subset of compounds, trains several machine learning regressors on fingerprint descriptors, and iteratively selects which compounds to dock next. It also includes an interactive dashboard and several utility modules, including a rescoring tool that re-evaluates user-selected top hits using a machine learning-based scoring function, paired with Shapley value analysis to improve interpretability. And finally, to wrap up this short list, which is definitely not exhaustive, let me mention SpaceDock, a structure-based computational method. It tackles make-on-demand libraries by docking about 150,000 reagents into a binding site and assembling compatible reagent pairs directly in 3D using a robust set of organic chemistry reactions, enabling the exploration of hundreds of billions of molecules that are likely to be synthesizable. That is it for today. This was a quick tour of just a few of the computational methods and tools presented at the meeting. Many excellent talks, posters, and applications to a wide range of therapeutic targets aren't covered here. So apologies to the authors I didn't mention, but I tried to keep the videos fairly short. If you'd like to learn more or become a member of the French Society of Chemoinformatics, I've added the link in the description. Thanks for watching, and until next time, take care.